inside the cell and induce gene expression, so it can increase the expression of genes that code for proteins like the sodium potassium ATPase. It can also increase the number and activity of the ENaC sodium channels, and it can also increase the secretion of acid or hydrogen ions from intercalated cells. The overall effect of aldosterone on the collecting duct is that it will increase sodium and water resorption. This will increase the effective circulating volume, and that's why aldosterone is part of the renin-angiotensin-aldosterone system. The job of that entire system is to respond to low perfusion pressure in the body and increase the circulating volume. Aldosterone will also increase potassium excretion, and that's why it is a hormone responsible for the regulation of serum potassium levels, and it will also promote acid excretion. Its release is stimulated mainly by angiotensin II and by high potassium. So angiotensin II is how the renin-angiotensin-aldosterone system stimulates aldosterone release. And then a second trigger for its release is a high potassium level in the serum. If you consume a high potassium diet, your body will begin releasing more aldosterone to bring the potassium level back down to normal. Finally, ACTH, which acts on the adrenal gland to promote cortisol release, can promote some aldosterone release, although this is a minor effect. Now we're going to talk about antidiuretic hormone, or ADH, and in order to understand how this hormone works, you need to understand the different sections of the nephron and their permeability to water. So the proximal tubule and the descending limb of Henle are permeable to water. Lots of water is reabsorbed in these segments, as we discussed earlier in this video. The ascending limb and the thick ascending limb and the distal tubule are impermeable to water. No water is reabsorbed in these portions of the nephron. And then the last section of the nephron is the collecting duct, shown here. The water permeability of this section is variable, and it all depends on antidiuretic hormone. Antidiuretic hormone is often called ADH. It is also sometimes called vasopressin because it can raise the blood pressure. It's a large peptide hormone. Note that it has a very different structure from the steroid hormone aldosterone. And what ADH does is it promotes free water retention and inhibits secretion of free water by the kidneys. There are two receptors for ADH in the body called V1 and V2. V1 is the receptor found peripherally that is responsible for ADH's vasoconstrictive effect that can raise blood pressure. The V2 receptors are found in the collecting duct, and they are responsible for the antidiuretic response of ADH, which we'll talk about in a minute. This hormone is released by the posterior pituitary gland, and there are two stimuli for release of ADH. You need to understand that ADH increases the amount of water in the body, so you want it to be released anytime the body needs more water. The major physiologic stimuli under normal circumstances is hyperosmolarity. So anytime the osmolarity of the serum goes up, ADH is released to dilute the osmolarity back down to normal. And on a normal day-to-day basis, this is the major stimuli for release of ADH. There is, however, a second stimuli that is important in many disease states, and that is volume loss. This is called the non-osmotic release of ADH because it does not depend on serum osmolarity. Basically, if your effective circulating volume gets low enough, that decrease in ECV will be detected by the pituitary gland, and it will release ADH no matter what the serum osmolarity is. This is a life-saving intervention. Basically, the body is detecting dangerously low circulating volume, and it's ignoring the serum osmolarity and just releasing ADH to try and conserve and hold on to water. The way that ADH stimulates water resorption is by acting on V2 receptors in the principal cells of the collecting ducts. These receptors are G protein linked with a cyclic AMP second messenger system, and once the V2 receptors are activated by ADH, an endosome is inserted into the cell membrane. I'll show you a picture of that in a second. The endosomes contain a water channel called aquaporin 2, and as a result of these channels being inserted into the principal cells, there's more permeability of those cells to water. So here's a picture of how this works. So ADH comes along and binds to a V2 receptor. That leads to the insertion of channels on the luminal side of principal cells called aquaporin 2 channels. Once more of these channels are in place, more water enters the cell, and then there are other aquaporin channels like 3 and 4 that are always active on the other side of the cell. So basically the amount of water being resorbed is controlled by the number of these aquaporin 2 channels, and the level and function of those aquaporin 2 channels is under the control of ADH. So the collecting duct can basically be completely permeable to water or completely impermeable to water. It all depends on the effects of ADH. So let's imagine that you are water deprived. For example, imagine you've been walking outside on a hot day, you haven't had any water to drink, and you've been sweating profusely. In this setting, your ADH level will get very high. That's because your plasma osmolarity is rising and it's being sensed by the pituitary gland, and ADH is being released. This is going to make the collecting duct here permeable to water through insertion of aquaporin channels. As a result, as tubular fluid moves down, it will equilibrate with the surrounding interstitial osmolarity. So that fluid osmolarity will rise to 600 in the outer medulla and to 1200 in the inner medulla. As a result, you will excrete very concentrated urine with an osmolarity of 1200 milliosms, and that's appropriate. You don't want to secrete much free water. You want to secrete mostly just solutes and hang on to all the free water you can. And that's what happens in water deprivation when ADH levels are high. In contrast, if you consume a high water intake, for example, if you chug 8 glasses of water, your ADH level will get very low. This will make the collecting duct impermeable to water. Now, as that tubular fluid moves down the collecting duct, some sodium chloride resorption continues to occur, driven by those pumps we talked about before in the principal cells. As a result, you will make urine that has a very low osmolarity, like 60. In other words, you are excreting mostly free water and very little solutes, and this is what you want to do. There's excess free water in the body, so you want to excrete urine that is very dilute and has a very low osmolarity. Earlier in this video, I told you that urea is a very important osmol for maintaining those osmotic gradients in the kidney along with sodium and chloride. I also mentioned earlier that the collecting duct is permeable to urea, and that's how urea gets into the interstitium. In particular, the collecting duct in the medulla, the medullary collecting duct, is permeable to urea. You should also be aware that urea reabsorption is linked to ADH. ADH increases the amount of urea resorption by the collecting duct and allows urea to enter the medullary interstitium to maintain those osmotic gradients, and then the urea is recycled by going back into the descending limb via transporters. This is easier to understand with a picture. So the first portion of the collecting duct shown here is permeable to water, but not to urea. This draws water out in the setting of high ADH levels. As a result, when you get down to this bottom half of the collecting duct, there is a very high concentration of urea found in the tubular fluid. That urea will then be absorbed along with water, and that adds urea to the interstitium. Transporters in the thin descending loop of Henle will pick the urea back up, put it back into the fluid where it'll go back around and then be recycled. This is called urea recycling in the kidney. But the key point here to remember is that urea is reabsorbed in the collecting duct, that it is influenced by ADH, and that urea is a very important osmol for maintaining those high osmotic gradients that you need out here in the medullary interstitium so that water can be reabsorbed. So here's a summary of the major functions of the collecting duct. It reabsorbs sodium and water, and this depends on ADH for water reabsorption, and it's under the influence of aldosterone for sodium reabsorption. The collecting duct also secretes potassium and acid, and this depends on aldosterone, and the collecting duct is also important for urea resorption. I'll finish this video with a couple of summary slides showing you where water and important solutes are reabsorbed in terms of portions of the nephron. So 2/3 of sodium is reabsorbed by the proximal tubule. Remember that the proximal tubule is the workhorse of the nephron. It reabsorbs most of all the solutes that are important in the body. You then reabsorb about 1/4 of the sodium in the thick ascending limb through those transporters we talked about, and the rest is reabsorbed in other portions of the nephron. So remember, for all the discussion that goes into aldosterone and ADH and all the reabsorption of sodium in the collecting duct, that's only a tiny portion of your sodium. Most of it is reabsorbed earlier, especially in the proximal tubule. And just like sodium, most of the water that the kidney reabsorbs is reabsorbed in the proximal tubule, about 15% in the thin descending limb, and the remainder can be reabsorbed in the collecting duct, but this all depends on whether the body wants to hold on or excrete free water, depending on the level of ADH. And that concludes our video on nephron physiology.